Hello everybody, welcome to the video demo for the Human Epigenome browser in Washington University in St. Louis. My name is Xin Zhou, I'm the developer. So in order to access our web service, please go to the WizHub website. Or you can find the link to this website from our lab website, which is here, and the link is here. Okay. So on the WizHub website, you can find the links to the web services that we built and maintained for the Roadmap Epigenomics project. And the Human Epigenome browser can be accessed by clicking this yellow link. Alright, so we opened up the Human Epigenome browser. Right now, this browser is best viewed with the Google Chrome web browser. It also works with Mozilla Firefox and Safari on the Mac, and it doesn't work with the Internet Explorer at this time. The major component of the Human Epigenome browser is this heat map in the center. It uses this heat map style to display the numerical values from the high throughput genome-wide DNA sequencing experiments, which in this case are all done for various types of epigenetic marks in the human cell types. So each row in this heat map is one track and correspond to one sequencing experiment. And when you mouse over, you can see the details, including the track name. And also on the horizontal axis is genomic coordinates. So the other important component of the Human Epigenome browser is this color block on the right of the genome heat map, which is metadata. So the metadata here is represented by the different colors for each of the track and for a number of attributes, and they have a lot of important functions for you to change the view and do the operations. So, and this is another topic in another separate video demo. And then right beneath the genome heat map, you can find the cyto bands of this region, and the position of this region in the entire chromosome can be found about the heat map in this image here, which is marked out by this blue box in this entire chromosome. And also you can find a link to the UCSC Genome Browser mirror, which we maintain locally. This mirror has all the tracks that are available on this Human Epigenome browser. By clicking this link, it will take you to the mirror and displays this identical region. Okay, so there's additional tracks that can be plotted under the genome heat map, so that user can look at the track scores in the context of various information, which is usually be the genomic features, and it can be something else, including the hypothesis test and so on. So on the bottom of the page is control panels. So this is navigation bars, and you can find the buttons. Click on it will opens up a tab on the right, which is a different or a group of related functions, and all of these will be described in the separate video demos. And the last component of the Human Epigenome browser is this toolbox. So you see that it's always on the same position of the page as if it floats, and you can click on the banner to move it, drag it to move it, so that it won't get into your view. It has some common functions that can be applied when you zoom, when you navigate, when you change the position you're viewing. You can click on this link to show more functions, more controls. And so in order to get help, the quickest way to get some very quick help is to click on this button on the top right corner of the page. By clicking it, many tooltips will pop out, indicating, describing very briefly how to use each component of the Human Epigenome browser. And click it again to dismiss. And also, in each tab of the control panels, and on the bottom you can find a very short text briefly describes, introduces how to use the about functions. And lastly, there's many important information on this help desk link. So this is our help desk. You can find the tutorials, video demos. So each button corresponds to one real demos, including this one, and source code. Alright, thank you very much.